So we've been talking about feedback loops, and I want to continue talking about feedback loops, and in particular talk about the implications of feedback loops for a very, very important idea in biology, which is called the Central Dogma of molecular biology. So what is that? Well, shortly after Watson and Crick in the late 40s and early 50s worked out the helical structure of DNA, Watson announced what they called the Central Dogma of molecular biology. And it's kind of taught, unfortunately, in a lot of classes straight from that today. So the central dogma is pretty simple to state. It says that DNA is the ruler and that instructions go from DNA to RNA, and that once transcribed, the instructions go from RNA to protein, and there is a unidirectional flow of information from DNA to RNA to protein. And that's what is generally called, as I picked this off the net, but you could pick 100 more like it. These are all illustrations of the central dogma, DNA to RNA to protein. The problem with this is that, number one, it is completely wrong. It is absolutely disproven. There are well-known counterexamples, which I want to talk about. And what's even worse is it's not just bad science, which it definitely is. It has given rise to some really bad social implications of science. It has given rise to racist science. It has given rise to some really bad science because of its obsession with genes and with the idea that genes are everything. And that isn't true. And it's produced a lot of bad science and a lot of bad social theory because people have not taken account of the incredibly important role of feedback relations. So the first blow to the doctrine of the central dogma of molecular biology was delivered by a very interesting person named Barbara Mcclintock. You probably never heard of her. So she got her PhD from Cornell in plant genetics in the 30s and 40s. And she started working with maize, you know, Indian corn, the kernels of the corn that have all the different colored little kernels. All of those color patterns are genetically determined. And so she was an experimentalist, and she took a big plot of land, ruled it with string into one square, little squares, and in this square, she crossed this with this. And in this square, she crossed this with the square of the cross of the this with that. And she did all of these experiments, and she spent years analyzing the data. Her analysis of the data led her inescapably to the conclusion that there were occasions on which RNA reprogrammed DNA. And when the Watson and Crick types were told that, they said, that's impossible, cannot happen. I just told you, the central dogma of molecular biology is DNA to RNA to protein. As one biologist told Barbara Mcclintock at the time, it's like the father to the mother to the children. There is a strict order of command. And Mcclintock stuck to it, and she said, no, this is what my data are telling me. And they told her again, they pulled out another metaphor. And a molecular biologist said to her, you know what it's like, Barbara? It's the boss and the secretary and the workers in the factory. And the boss dictates the letter, and the secretary types the letter, that's transcription. Secretary types the letter, and then takes the letter to the workers in the factory who make the protein according to the instructions of the boss as typed by the secretary. Now, Barbara, we know that there can be bad secretaries. Viruses can mistranslate the DNA. But Barbara, the secretary does not reprogram the boss. There can be mistyped letters, but the boss himself is never altered. The DNA is immutable. And she said, no, my data say there are mutable elements in DNA and that the RNA in the right circumstances can push them around. So her work got published, as you see. She published in 1950 in PNAS. But the work was totally ignored. 20 years later, in 1975, or before 1975, Howard Tiemann and another group of molecular biologists, this was now a subject, found that certain viruses could carry out what they called reverse transcription. Now, you know what transcription is. It's the path from DNA to RNA. And reverse transcription is the path from RNA to DNA. And in particular, reverse transcription may ring a bell with you if you've studied HIV at all. You know that the way HIV takes over the cell machinery when it infects you is with a protein called HIV reverse transcriptase, which is how HIV, which is an RNA virus, alters your DNA by the process of reverse transcription. So everyone said, oh, this is a fantastic discovery, this Nobel Prize. And then someone said, you know, there was a crazy lady, that crazy lady at Cold Spring Harbor, Barbara Mcclintock, said this 20 years ago. So they looked it up, and sure enough, she had said it 20 years ago. And so she shared in the Nobel Prize that was given to Tiemann and the others for discovering this process by which RNA reprograms DNA. So that is just a counterexample to the central dogma. And by the way, I have to say something. I approached one of our esteemed biology instructors who was teaching the central dogma, and I said, hey, John, not his real name, I said, hey, John, you really have to stop saying that because it isn't true. And take, for example, Mcclintock and Tiemann and the Nobel Prize for reverse transcriptase. And he said to me, he said, yeah, the central dogma is not 100% correct. It's a simplification that we have to teach at this level. Well, I don't believe that. I don't believe it's ever right to teach stuff that's just wrong. And the central dogma is just wrong. So let's go on to look at some more cases in which it turned out to be wrong. Again, shortly after Watson and Crick, some very good French biologists, Monod, Jacob, started to ask a question. For example, how do we get repeated expression of a gene, except by making the gene work again and again and again? Is there some feedback present? And they said, oh, yes. And this was the work, incidentally, that led to the Lac Operon, which we have just been discussing. They said genes have responsive elements that are not coding for proteins, but are rather the receptacle for protein action. So the gene makes a protein, and the protein comes back and binds to the responsive element. In the specific case they were talking about, it was the positive feedback that constitutes the action of the Lac Operon. And so this positive feedback loop, they pointed out, can turn on expression for a long time after a single stimulus. And that's a great property to be able to have. And what they showed was that this positive feedback mechanism, which was from DNA to protein back to DNA, is a very important feedback loop that produces important phenomena in the production of this protein. Jim Farrell, who is at Stanford, wrote a very nice paper recently, or more recently, in which, and I want you to look at the title, Self-Perpetuating States in Signal Transduction. Positive feedback, double negative feedback, and bistability. So this is all stuff that is beginning to ring a bell. Bistability is something we've been talking about. And what Farrell points out in this article is exactly what we've been saying, which is there are all sorts of system properties that you want from a gene regulation network, like self-perpetuating states, like a permanent effect from a transient stimulus. And how does the system do that? So as Farrell said, talking about Monod and Jacob, they drew diagrams. They weren't writing differential equations, but they were drawing diagrams that represented positive feedback loops. And these positive feedback loops included the Lac Operon. And this was the work for which they won the Nobel Prize. And they were able to show that this positive feedback loop here was embodied in the E. coli cell, and by means of this sigmoidal positive feedback here, produced this biological switch. So the question that they addressed is, how do you make if given the central dogma, how do you make a biological switch? And the answer is, you can't. And so therefore, to have a biological switch, we must be incorporating feedback mechanisms that turn things on and off. And as you know, and this is now just repetition, we've talked about this, we have in this system the three equilibrium points of the bistable system and the existence of a biological switch. And so saddle node bifurcation, which is what we talked about in the context of the Lac Operon, means that in this region, for example, we have a stable, an unstable, and a stable equilibrium point, and a robust biological switch which can turn on expression.